this photo shows a picture of Arya when she started in the MSTP program, which is the combined MD-PhD program in the College of Medicine. And we were really thrilled to have her because she was really an outstanding student who did her undergraduate studies at William and Mary, and then came and joined the MSTP program. And you can imagine how delighted I was when she decided to rotate with me and then join my lab for her PhD. Arya is everything that we thought she would be. She's hardworking, she's extremely intelligent, and you know, she's going to be a terrific physician scientist in the future, and she's already a junior one now. She takes everything really seriously. She's a serious overachiever, and I'll just show you that this is true even without So I can always count on Arya to go above and beyond when it comes to anything. And you're going to see that in her thesis presentation today. But on a personal note, she's also just a terrific person. She's got a great sense of humor. It has been my absolute pleasure to be her mentor and to have her in the lab for the last almost 4 years. And so with no further delay, let's go on to the main part of this show. Arya.
Thank you. Yes. I hope that'll go. Okay, perfect. Okay, hi everybody. Thank you so much for being here today. Oh, it's very nerve-wracking, but we can do this. My name is Arya Zaha. So today I'll be going over my thesis project entitled The Longitudinal Skin Microbiome in Pediatric Atopic Dermatitis. So, okay, here is the outline for today. I will be going over the background, a brief overview of the thesis part 1, which focuses on temporal Staph aureus colonization, part 2, which focuses on the differences between contact plate versus skin tape strips and sequencing, part 3, which focuses on longitudinal skin metagenomic shock and sequencing data. Finally, I'll end with the conclusions, take any questions, and then go on to my acknowledgements. So let's start with the background. Atopic dermatitis, or AD, is a chronic inflammatory skin condition. It's typically localized on the cheeks and flexure surfaces. So this is my niece, she has AD. Both of my nieces have AD. They're here today for demonstration purposes. So AD is typically diagnosed in the first 6 months to first year of life, and is prevalent in about 15 to 20% of children. The prevalence is increasing, especially in developed countries. AD has one of the highest burdens of all non-fatal diseases and the highest burden of all cutaneous diseases. So it has negative effects on quality of life due to itch and sleep disturbances. It also has effects on psychological and emotional health due to negative social stigma, anxiety, depression, and then it also has a huge financial burden due to having to buy soap, detergents, decreased productivity, etc. AD is also the initial disease in the sequential progression of allergic conditions known as the atopic march that share common predisposing factors and immunological features. So the atopic march includes AD, followed by food allergy, asthma, and allergic rhinitis. And while not all children follow this march, having one of the allergic conditions increases the risk for developing others. So it's critical that we study AD to help potentially prevent the progression to these other atopic diseases. So AD is characterized by 3 major components shown here. So the first one is local and systemic immunological dysfunction. The second is barrier defects, typically characterized by decreased expression of skin structural proteins such as filaggrin. And then finally, there's also increased exposure to environmental factors such as allergens or microbes. It's really important to remember that these things don't exist. And in terms of bacteria, they can be commensal, so typically they reside on skin and they don't cause harm to the skin, or they can be pathogenic bacteria such as Staph aureus. A lot of different factors, including the physiological site, the age of the microbiome in adults is stable across time, but the composition is determined largely by the physiology of the skin site. So for example, your oily areas such as your face, your chest, and back contain sebaceous glands that produce sebum and that house lipofilament delivery, so whether you're born via C-section or vaginally. And then bacterial communities at different skin sites actually be pretty diverse populations. Some of them might have Staph aureus as shown with the red, whereas in AD skin, as shown on the right here, there's microbial There's reduced diversity. We also see increased Staph aureus, and I'll be going over all of these. So part one is looking at the temporal relationship between skin barrier defects and Staph aureus colonization and how that affects. Or it's the first U.S.-based early life AD core. It's also a diverse cohort that reflects a few things. We look at filaggrin, which is a skin structural protein, the expression of filaggrin. We also, to show two different sampling methods, this is actually also my niece, my other niece. She's almost 5 now, she's sitting at the front, but she was not an MCASH participant. But we utilize contact plates directly on the skin surface to grow bacteria, specifically Staph aureus. And then we can isolate and freezing all the DNA that we capture, and we're able to answer two important questions that we could not do using the contact plates. The first one is asking what is there. So we can look at the comprehensive microbiome instead of looking at just one microbe such as Staph aureus. And then between skin filaggrin expression and Staph aureus colonization. So this was the basis for our first paper, which was published 2 years ago. I'll be going over the results very briefly. Higher sporad, more severe AD, more allergen sensitization. And we also see a decreasing trajectory of filaggrin expression from visit 1 to visit 2. Interestingly, what we find is that the low filaggrin expression actually mediates the colonization that we see at visit 2. And then conversely, if there's no colonization at visit 1, regardless of colonization at visit, if there's no colonization at visit 2, regardless of colonization at visit 1, this is associated with a lower sporad, milder AD, less allergen sensitization, and then we see a steady expression of filaggrin. Thus, these are novel findings where we start to understand this temporal relationship, where we observe that skin barrier dysfunction may actually be mediating further pathogenic microbial colonization. So in part one, we looked at Staph aureus colonization using contact plates, but like I had mentioned before, we also utilized skin tape strips to detect Staph aureus and MPatch. So in part two, we wanted to address the following gaps as rationale for our studies. So currently, there's no gold standard in skin microbiome sampling and analysis, leading to variability in microbial detection and interpretation. And there's also no consensus on a method that reliably correlates detection at each visit for 328 participants. Given the nature of a longitudinal and ongoing study like MPatch, some of these matched samples could be from the same individual at multiple time points. Thus, for the analysis that I'll be sharing today, all the duplicate individuals, 52 individuals with 22% that had it detected by only sequencing, whereas 38 or around 12% had it detected by only contact plates. And then this is looking at it a little bit differently here. So there's 328 individuals. Out of 328, 105 had samples detected through sequencing, had Staph aureus detected through sequencing. And then out of the 105, 72 or 69% was not detected by contact plates. Meanwhile, out of the 328, 71 samples were detected by contact plate. Out of the 71, 38 or 54% were not detected by skin tapes. So this finding was really interesting to us because this tells us that contact plates and skin tape strips have that sporad, starting from the left here, which is, as I mentioned, a measure of AD severity, was higher in the double positive group compared to the other groups. Correspondingly, there was a larger percentage of children who were moderate to severe AD. In the double positive group, there's also a higher percentage that were sensitized. And then we also observed that they were sensitized to their contact plate, and skin tape samples have to have come from either a dry site or a sebaceous site. So both of them have to come from the same type of site. And here we see very similar trends, even though we have less samples here with increased sporad, higher sensitization load, and a similar trend here where we see that the total serum function is higher in the contact plate only positive groups and the double positive groups. So this is telling us that the double positive detection was associated with worse AD outcomes, even with samples from matched dry or sebaceous sites. We decided to look further into this trend of staph-specific IgE. And here I'm showing a similar study where for a subset of the samples, we looked at the detectable Staphylococcus enterotoxin A-specific IgE as a measure of Staph aureus and a measure of Staph aureus biological relevance. So we see an interesting trend here that kind of mimics what we saw with higher total serum IgE in the double positive groups. Staph aureus detection was associated with increased severity and sensitization. And I didn't show the data here, but there was also a higher percentage of children with allergic rhinitis in the double positive group, and also a higher asthma risk in non-Black children in the double positive group. Allergies and MPASH, we wanted to explore beyond Staph aureus to look at the comprehensive microbial communities over time in children. In previous literature examining the skin microbiome, either on infants, adults, or healthy individuals, they don't examine it on the children that are aged 1 through 10, 1 through 13, and I provided some examples of recent studies that look at the skin microbiome in these specific groups, so infants, adults. When we look at the microbiome in infancy or adulthood, we fail to examine this very critical window of dynamic microbial patterns that may help inform changes related to atopic development. We used age instead because the age range within each visit was pretty wide, and you can see the samples at each age range in this table right here. So I just want to explain some important concepts. Each of these species occupy half and half, so 50% of the community. So now you're increasing how equally distributed the species are. That's called evenness. We can also increase the richness, which means that we can increase the number of species that are present in that sample, so as you can see here. And then you can kind of do a combination of both, where you increase richness and evenness, where now you have more species, but now they're also more equally distributed in the community. This is the concept of alpha diversity. You can use evenness or richness as diversity measures, or you can also use a combination of the two. So for example, we use something called Shannon diversity that takes into account both richness and evenness of the community. So alpha diversity is specific to one community. We can also compare two different communities, right? So we can use another measure called beta diversity. So you can calculate the similarity distances between two communities to estimate how close they are to each other in species composition. Imagine you have a lot of samples. You can plot this on a dimension reductionality plot such as this, and this allows you to visualize how similar or dissimilar the communities are. And then you can also group them by different variables that you're interested in. So for example, I have it grouped here by age, and we can say that age explains a certain percentage of the variance in species composition. So now that we have this information, let's both Shannon diversity and observed richness, we see lower values in older children compared to younger groups, with a decreasing trend as the children get older. So these results reinforce or indicate that there are fewer species as the children get older, and that a few species.
See a hand up there. Sorry, Alex, you want to go first?
Interested in the dysbiosis, and it seems to me that the diversity is decreasing with age, but not necessarily. It's always like predominance of Staph aureus that's actually dictating severity. That is in the skin, but the dysbiosis of the gut is in favor of cluster 5 for protection. Correct? So cluster 5 would be less progressive, essentially. Loads of probiotics. So according to you, which is more important, dysbiosis of the skin or the gut? And I feel like because we're talking about atopic dermatitis and the insult is from out in, so whenever we have aureus in the skin, it's a strong hit essentially. But we can still play at the dysbiosis in the gut to actually protect. Which one will win?
Oh, that's a good question. I mean, so allergic diseases, like the foundation of allergic diseases can be either from, there's two theories for both, right? So there's either from epicutaneous sensitization, or it can be through the gut where, in Western diets we're getting less fiber, more fat, there's less production of short-chain fatty acids, and that also leads to an allergic phenotype. I mean, I have to choose the skin, just doesn't work on the skin, but it is cool that we're seeing gut microbes on the skin. At least we're picking up DNA from the gut microbes, right? We're not sure if they actually live there or not. So, I think to that end, there's a lot of interplay between the two in allergic diseases.
Yeah.
Thank you.
Go, go, go.
Congratulations.
Thank you so much. Oh my gosh. Yep. Thank you. Did she cry?
Can you do it again? This is dartboard.
And that's also an doctor. Doctor.
That was very loud. Alright. Nobody look at me.
How bad was it? Was it bad?
No, not at all. Good.
Wait a minute. Nanny. It's Ara. Okay.
Well, she earned it, I'll tell you that. She did a great job on the presentation. She handled the questions like a pro.